Hello, everybody. My name is Marie, and my presentation is going to be on carbon dioxide transport in the blood and the Haldane effect. And I'd like to begin with the idea of total pressure and partial pressure. Total pressure is the sum of all the partial pressures of gases within a system. And if we look here on the left, we can see the air that we are breathing in, mostly nitrogen, some oxygen, very little CO2, and some other gases. And then, of course, water is variable. And those gases exert a certain amount of pressure. So the alveolar air, let's say, is 568 millimeters of mercury for nitrogen, 105 millimeters of mercury for oxygen, 40 millimeters of mercury for CO2. But if we look in the systems, our pulmonary system and systemic system, we can see that there are partial pressures of these gases, and the pressure exerted by a particular gas is directly proportional to the percentage of that gas in the total air mixture. So when we look at the oxygenated blood that's coming from the pulmonary system going into the systemic arteries, we can see that oxygen is much higher than CO2 at this point, but not so much over here. Once the body tissues have used up and metabolized and done cellular respiration, they're producing CO2, and that's exerting a higher pressure here. And this is important because there is always a gradient. The systemic partial pressures never really equilibrate between the oxygen and CO2 because these cells are constantly using the oxygen and producing CO2. So the tissue oxygen is slightly less than the oxygen of the blood, leaving the systemic capillaries. The tissue CO2 is always slightly higher than the systemic venous CO2. Carbon dioxide is transported in three ways. It can be physically dissolved, bound to hemoglobin, or as bicarbonate. Carbon dioxide is more soluble in blood plasma than, say, oxygen, and 10% of the carbon dioxide is in this form, just physically dissolved. 30% of the carbon dioxide is bound to hemoglobin in a form called carbaminohemoglobin, and this is where carbon dioxide has an affinity for the globin portion as opposed to the heme portion of the erythrocyte. Oxygen has a greater affinity for the heme, or I should say, heme has a greater affinity for the oxygen, whereas carbon dioxide binds to the globin portion. And if we look at this diagram here, we can see these three ways that CO2 travels, either dissolved or as the carbaminohemoglobin. And then we can see how the tissue cells are producing this CO2 and oxygen is being dumped off into the tissue cells. In the alveoli, this is where oxygen is abundant and it's traveling into the red blood cells. And here is CO2 that's dissolved and CO2 that is within the red blood cells being dumped off into the alveoli. The third way is as bicarbonate. And I'm going to jump ahead here. And the formation of bicarbonate is really the most abundant way that carbon dioxide is transported in our blood. And carbon dioxide forms with water carbonic acid and then bicarbonate and hydrogen ions. And it does this slowly in the plasma, but rather quickly in the red blood cells because of this enzyme called carbonic anhydrase. And sometimes it goes so quickly that it bypasses this carbonic acid portion and goes directly to forming bicarbonate. And if we look in the diagram here, we can see that this carbon dioxide not only can be dissolved in the plasma, combining with the hemoglobin, but here forms the bicarbonate. So again, the three ways that CO2 is traveling. Hydrogen and bicarbonate start to accumulate in the red blood cells, and there is a carrier within erythrocytes that is the bicarbonate chloride carrier, and it allows for the passive diffusion of bicarbonate out of the red blood cells, and there's an electrical equilibrium gradient that is constantly shifting, and chlorine actually moves into the red blood cells, and this is called the chloride shift. So, what happens to these hydrogen ions here? If there's a buildup of hydrogen ions, then we have the pH dropping and acidosis, or the lowering of the pH. And what happens is hemoglobin actually will take up these hydrogen ions. If this didn't happen, then the venous blood would be much more acidic than the arterial blood. And it's a very efficient exchange process where hemoglobin is unloading oxygen as the carbon dioxide and hydrogen are increasing, and it's being picked up, and it's picking up the hydrogen ions. And we can see that in the third portion of this diagram here, in the red blood cells, the bicarbonate being made, and there are the hydrogen ions. The opposite holds true in the alveoli. So all of this is known as the Haldane effect. And that is the idea that hemoglobin has this greater affinity for hydrogen as opposed to the hemoglobin when it has oxygen does. And it's unloading this oxygen, which facilitates the picking up of the carbon dioxide and hydrogen ions. And all of this was first established by John Scott Haldane, who is known as the father of oxygen therapy. And he was a very interesting scientist. He was very interested in connecting the theoretical sciences with the applied scientists. So he was the first person to really describe the fact that the rate of respiration and oxygen uptake into the bloodstream are both controlled by the carbon dioxide content of the air being inhaled. So we inhale because of this buildup of CO2. Remember when you were little, we all tried to hold our breaths for as long as we could, and you can't because your brain stimuli will tell you to breathe because of the CO2 buildup. I included some pictures here of some respiratory devices. This was on a Mount Everest expedition in 1922. And again, Haldane was one of the first people to figure out respirators. He did work in the mines and caves, and in 1896 he demonstrated the presence of carbon monoxide in caves. He actually climbed Pikes Peak in Colorado and did an experiment at 4,000 feet to figure out the air quality and the breathing ability of climbers. He was one of the first people to figure out decompression charts for divers. And in World War I, he was recruited by the British military for medical research to figure out respirators because of the gases that were being used on the soldiers. At that point, they were still using cotton wrapped in gauze to help to keep themselves alive. There can be problems with carbon dioxide transport when it's not working properly, and hypercapnia and hypocapnia are two conditions. Hypercapnia is too much carbon dioxide in the blood. It increases the pH levels and causes acidosis. A clinician would check for arterial blood gas of carbon dioxide, and if it's greater than 42 millimeters of mercury, then that's considered hypercapnia. Hypocapnia, on the other hand, is low CO2, and that's generally well tolerated. And here is a study on carbonic anhydrase 9, a study that was trying to determine if microvascular endothelial cells play a role in pH regulation and angiogenesis. And angiogenesis is the formation of new blood vessels, and pH levels have to be at a certain level. And what they did was they took two kinds of cells. microvascular cells and pulmonary arterial endothelial cells. And they grew these cells with the idea that the microvascular cells, they were going to knock out this carbonic anhydrase 9 enzyme. So their hypothesis was that the enzyme is critical to pulmonary microvascular endothelial cell and angiogenesis because of its role in regulating pH. So what is its ability to regulate pH? So they did this study with rat cells, and they had knockout endothelial cells, normal endothelial cells, and then normal arterial cells. And through many different methods, growing the cells, monitoring the pH, doing a Western blot, they generated the carbonic anhydrase 9 depleted cells by way of CRISPR-Cas9 and did cytotoxicity assays and statistics. And what they found was that indeed carbonic anhydrase is important in regulating pH extracellularly. So when we look at their results here, there are 3 graphs, A, B, and E, that show variations in treatments of cells, and they monitor the pH over a period of time. And it did appear that the microvascular endothelial cells were not affected by the decrease in pH. Over here, we can see the growth of the cells over a period of about 7 days. I think it was a little bit longer than that too. And these are various buffers for each of the pH systems. And we can see that the cells, the microvascular cells were growing very nicely at these pHs. Not until it hit about 6.2 did they notice that the cells really dropped off. However, the arterial cells were affected and did not grow as well. Over here is a Western blot, and it does appear that the microvascular cells had a high expression of the carbonic anhydrase 9. They have a higher expression of it even in the acetic medias. So carbonic anhydrase 9 is an important determinant of pH regulation in the microvascular cells and in angiogenesis. They think that this may give us a better understanding of the mechanisms of acidosis-induced lung injury and hopefully to develop therapeutic strategies. Thank you for your